Okay, I can
Thank you.
introduce myself. So, good. Well, it's not good morning. It's morning here, but good afternoon, everyone. I would really have loved to been at UCLA to give this in person to meet the patients and the families and other researchers, but unfortunately, I committed to this conference quite a while ago. So I'm presenting early in the morning in Queensland, Australia. Um, so you probably can guess already that my accent is from Australia. So um, I'm a researcher at Yale University, researching into the genetics of a lot of different neuromuscular diseases, and also I've been working with the NDF since the around about the start of this year. So coming on board when the whole genome sequencing projects, as Maduri would have had presented, started to be launched, and the commitment to do some of the whole genome sequencing. And so the focus of today's talk is to talk about the road to gene therapy approaches and all the things that actually have to happen either at the same time or before we transition to gene therapy. And the reason why is that every neuromuscular disease is different and therefore has a different set of challenges, and also a different set of challenges in understanding the mechanism behind how it actually causes disease. And a lot of these genes, we're still trying to understand fully how they work. And the reason why we need to know how they fully work is then to then say if we actually gave patients a replacement of that gene, whether that would actually work too. So before that, I'll tell you a little bit about myself. So for those who have attended webinars in the past, this may be a bit boring, but it would be a good refresher anyway. So I'll just tell you about my journey as a scientist and also as a patient. So I first started off in the IT industry, worked in IBM for 3.5 years, but left in 2004. And then I did a bioinformatics and physiology degree and completed in 2008. So all this is in Australia. I finished my PhD at the University of Sydney in genetics in 2012. And then I wanted to go to Boston in the United States because with the one goal of reducing the time that families have to wait for a definitive genetic diagnosis. And it's very important to have that genetic diagnosis, as that's the first step in the road to any therapies for patients in their journey. And a lot of patients go on this quite a long journey, and I wanted to work on technologies to reduce that journey time. And so during that training, I worked on, had a good fortune in working on one of the largest projects at the time, the Exome Aggregation Consortia project. And this project was aimed at creating a reference panel of relatively healthy people. So when we discover mutations in neuromuscular disease patients, we have a better ability to interpret if it's likely to cause disease or not. And that applies to GNE myopathy too. When we see rare variants in GNE, we want to know, is there anyone else in the population? And if it's too common, it's unlikely to cause GNE myopathy. I also worked on other rare muscle disease cohorts, collaborating with a lot of people around the world. And I think that's why I think it's now made it a lot easier for me to work with some of the collaborators because of some of the work that I've worked on. And also, they're normally collaborators of collaborators. So although I haven't worked directly with all the GNE myopathy researchers, I've always worked with some of their collaborators also. So that's working with exomes, genomes, and transcriptomes. So genomes and transcriptomes, I'll talk a little bit later in this presentation, and its importance in understanding GNE myopathy, and also understanding if we had that information, if gene therapy or other future therapies are actually doing, actually providing short-term benefits along with the long-term benefits. And lastly, before leaving for Yale, I led an analysis group at the Center of Mendelian Genomics, where its main aim was to discover novel disease genes in patients. So that would be helpful for neuromuscular disease patients. About 50% of them don't have a definitive genetic diagnosis. And also on the Gabriella Miller Kids First, which is a large genome sequencing project in undiagnosed patients. So which has given me a great deal of experience of working with whole genome sequencing data and all the challenges, both scientific challenges and also computational challenges with working with data sets that large. The reason why I care about gene therapy and the road towards gene therapy is that I'm actually a patient myself. I have limb-girdle muscular dystrophy, and that picture of my legs were taken about 6 years ago when my legs weren't as wasted. So they're probably a lot more wasted now. So I know the challenges that GNE myopathy patients have. So the onset for me was around 19 years of age. I had difficulties going upstairs, getting up off the ground. And nowadays, I don't travel so well. If I need to go long distances, I need a wheelchair and things like that. So for me, I can empathize with a lot of the people in the audience. And so I'm not just a researcher, but I'm also very keen in not just understanding the disease, but how that can actually translate to actually finding therapies for patients to both stop the devastating wasting that happens in muscle disease and also hopefully stabilize the condition and one day also reverse some of the muscle wasting that has happened. So the current status of gene therapy based on what's published, unfortunately I can't present unpublished information, is a lot of hard work by Stella and her colleagues and collaborators in showing in a wild-type mouse that AA delivery of GNE myopathy is both efficient, so it gets to a lot of the mouse muscle, and it's also safe. So these are two important things because before trying this, it was thought that the lack of sialic acid at the cell surface would affect the virus from actually doing its thing, that is entering the cell. But it's been shown in this set of experiments that it doesn't actually affect that. It's also safe. So the thought is if you overexpress GNE, the gene itself or the protein or the enzyme, that it may not be safe if we're overexpressing it, but these mice seem to be quite healthy. But one thing I want to clarify, these are actually healthy mice that were injected into. So it was only looking at efficiency and safety. And the reason why is currently, as far as I'm aware, there is no reliable mouse model. And this is not just a challenge in GNE myopathy, but in a lot of the muscular dystrophy mice, that some of them display some features of muscle disease, but not all features. And it's been pretty tricky with GNE myopathy to create a mouse model that reliably creates the muscle wasting pattern that we see in the patients, because this is sort of like the hallmark feature of the disease and something that we actually want to stabilize. And so with that, what I have in red is we still need to show effective delivery that slows or stops the disease progression. And we need a much reliable mouse model to show that. The other thing to note is that AAV, in terms of the immunogenic response, that once you've actually received it once, sometimes the second and third time it will not be as effective because your body has actually recognized that it's seen in the AAV. And in fact, that some proportion of the population, because a lot of these AAVs are naturally occurring also in the population, that some people have been exposed to it already. So it's just something to keep note that if we do want to progress onto gene therapy, that we need to be very careful that we know it's going to be effective, because safe is not what people want. Efficient is not what people want. People want something that's effective, that actually stops the condition, because just having GNE in there or anything that we think would be a good therapy is great, is a good first step, but a better step would be if it stopped or halt the progression. So in terms of translation of results to patients, some of these are the things that I see as important goals, high-level goals. So first is the genetic characterization of patients. So some of that Madhuri would have talked about, so hopefully it isn't too much of an overlap and a bore for the people in the audience. The importance of natural history studies. So I know Ultragenics and also the NIH have done some great work in terms of creating natural history studies. And this is important in terms of reducing the number of placebos we need in some of these clinical trials, that we can use what we see as the natural progression of the disease as a benchmark to compare against, rather than using too many placebos. The next one I already touched upon is animal models with a reproducible diseased muscle. And that way, when we're actually testing the effectiveness of any therapy, we can see if it halts or reverses that muscle condition that we see. Next is patient cells. So I know that individually, a lot of the collaborators around the world, through their hard work, have worked on patient cells. But what we'd like to do is, and I'll talk about it later on in this presentation, is also establish more patient cells, and to do it in a sort of like a consistent way and also in an engaging way. So we would like, if we got patient cells from patients in the audience during this symposium, is to engage with them afterwards, to show what we're actually doing with the cells and things like that, and not have people wait until there's a publication or stuff like that. And it's great to have the mechanism of the NDF and their webinars as a way of communicating some of those stuff. So I'll talk about that towards the end of what we could do if we had patient cells. So it's not to say we don't already have patient cells from patients, because collaborators around the world have done that. But I'd like to introduce some of the new technologies that we could do with their patient cells. Next is to develop a robust biomarker to measure the effectiveness of these therapies. So it's very important to have these biomarkers, mainly because the progression of the disease is very slow comparatively to other neuromuscular diseases. And so we have to have ways of measuring, when a therapy has been applied, the short-term benefits also, not just the long-term benefits. So we have that sort of like warm and fuzzy feeling that things are actually working. So there are two projects that will complement the road towards gene therapy. The first is the whole genome sequencing of patients and their families. And the reason why is, firstly, we have the right target in each patient. And it's very important to understand this and to fully genetically characterize the patients. But to clarify, this is not a diagnostic effort. For patients for this whole genome sequencing effort, they should have already been diagnosed with GNE myopathy with the mutation already known. It's just to understand, are there other things that are contributing? The other thing is, by doing genome sequencing, we may be able to ask, are there other targets that may contribute to the disease? And why this is important is that it actually gives us alternative things that we can actually look into in terms of developing therapies instead of just the GNE myopathy gene itself. And this has been beneficial in other forms of neuromuscular disease, to go after other targets that are so like directly related to the disease gene itself. The next is initiative that Angela, my wife, and also a consultant for NDF, is very passionate about. And I'm going to present some of this also in the second half of the presentation, on why it's important to have a GNE myopathy patient biobank. And it's to develop robust genetic biomarkers. So the emphasis on genetic, and I'll go through sort of like the differences between genetic and non-genetic biomarkers. Patient cell lines to understand these better, so I've already touched upon that. And proof of concept on using different therapy approaches. So by having these biomarkers and having cells, we can try a lot of different things without bothering patients first. So cool. So I'm just going to touch upon, you've probably seen slides on this throughout the whole symposium, even after when I present, is that GNE myopathy, you can see one of the hallmark features are the inclusion bodies that we see here in the patients. And so there is both a genetic diagnosis and also a clinical diagnosis. The clinical diagnosis would be a clinician saying you have the overlapping features of GNE myopathy, you have the hallmark features, including inclusion bodies, but a genetic diagnosis is a lot more definitive. That's actually looking at the GNE myopathy gene itself and finding the mutations in your DNA. And because it's a recessive condition, we're actually looking for two mutations in the DNA. One, so either can be homozygous, so it's the same mutation twice, which a lot of patients do have with the founder mutation, or heterozygous, so two different mutations. So there are a few different ways that people come about a genetic diagnosis in GNE myopathy, depending on the time period. So before the use of next generation sequencing technologies, which I think Madhuri would have touched upon in achieving whole genome sequencing and also exome sequencing, before that, we did just direct screening on genes that we suspected that was actually causing disease, or doing a gene candidate approach based on the clinical features the patient has. So some patients have had direct screening of the GNE gene. Some of them have been discovered by screening a set of genes that we know already cause neuromuscular diseases, and that's how the two mutations in GNE myopathy have been detected. Next would be exome sequencing, and then next would be genome sequencing. But a lot of patients, I assume, would have been diagnosed genetically by the first two methods. And the exome is, just to clarify, is about a 1.5% of the slice of the whole genome apple. And this is the coding regions, or the regions that we that have the instructions to make protein. And this is the region that we understand the best, while the rest of the whole genome is a lot harder and difficult to interpret, but we're getting a lot better. And that's why it's important now to sort of transition to genome sequencing. As we get better, we also get the exome, the protein coding regions of the genes. So the genetic diagnosis, the challenges are the clinical presentation can be caused by many different disease genes. So a lot of diseases may look like GNE myopathy in terms of how the patient looks like, the struggles they have with day-to-day things, and also the muscle biopsy and some of the degeneration we see in the muscle biopsy. And also many different genes have the same clinical presentation. So it's a challenge of, did we get the right gene? And we know that each person has about 20,000 genetic variants in their genes that encode for protein. And 100 of them are very rare in the healthy population. And this is some of the work that I was doing when I was in Boston, trying to create resources and tools to actually sift through that needle in the haystack to find what mutation is actually causing disease in the patient. And the hardest thing to know, to deal with, is that the majority of rare variants do not actually cause a disease, and that they're just benign and they just happen to be there by chance. And that's the challenge of genetic diagnosis. So the other challenge is direct screening of the GNE gene, or screening of genes that cause neuromuscular disease, is like looking under the lamppost. As you can see, this person here looking for their keys under the lamppost. Sometimes we always wonder, what is outside that lamppost? Are there other genes outside that could actually contribute to a patient's disease, or just challenge the diagnosis? But most of the times we're asking the question, could there be other mutations that could either be making the disease less severe or actually more severe? But we will only know if we look outside the lamppost, which is the equivalent of doing either genome sequencing or exome sequencing. So the challenge of, however, of checking all genes is the difficulty in interpreting all the rare variants that are discovered. So just because they're rare, like I said, it doesn't mean that they may have an impact. And that's why we need a lot of patient samples also to see some of these patterns, either in genes or in the particular rare variants that we see in itself. And by having more numbers, we can have that better ability to interpret that. So with that, these are the aims of the genome sequencing project is, first of all, to confirm again the GNE mutation, that the mutations that are in the genetic reports are indeed what we see in the patient. This is just due diligence. We shouldn't see any surprises here, but it's one of the first goals. The first is to identify additional genetic contributors to the GNE disease. So in terms of severity, onset, and differences in conditions. So by creating the right kind of cohort, we can. So the challenge however, of checking all genes is the difficulty in interpreting all the rare variants that are discovered. So just because they're rare, like I said, it doesn't mean that they may have an impact. And that's why we need a lot of patient samples also to see some of these patterns, either in genes or in the particular rare variants that we see in itself. And by having more numbers, we can have that better ability to interpret that. So with that, these are the aims of the genome sequencing project. Is, first of all, to confirm again the GNE mutation, that the mutations that are in the genetic reports are indeed what we see in the patient. This is just due diligence. We shouldn't see any surprises here, but it's one of the first goals. The first is to identify additional genetic contributors to the GNE disease. So in terms of severity, onset, and differences in conditions. So by creating the right kind of cohort, we can ask these questions. We may not be well powered to have significant results, but we have to start somewhere, and then we can go and validate in other samples if we make that discovery. And as I touched upon, to identify other genes or pathways to target by looking at other mutations outside of GNE myopathy that we see in patients. It also allows us to prioritize the best set of patients for clinical trials based on the genetic characterization that we've made. And lastly, and it will further our understanding of disease and strengthen scientific collaboration consortia. So in order to build this cohort, we have to work with a lot of the scientists from around the world. And by working together on a project, it will build a lot of goodwill and also sort of like strengthen the relationship by everyone contributing and working together with this shared data. And I've seen this collaboration work for other rare disease consortia, and I think it's a way of future in terms of working together. So in terms of the patient cohort and recruitment, the NDF has done a fantastic good job of reaching out to the patients. So in the next few weeks, I hope to have time to actually review it and maybe have the NDF reach out to get some additional information so we can actually review it and see which ones would actually fit in the inclusion criteria. The next is strategic collaboration. So I've reached out to many of the major collaborators around the world who have these cohorts and to pick a set from them. And so this is the progress today. We're currently finalizing inclusion criteria of which patients would be the most informative and which sort of like design and which family members should we have to actually have the most informative set. We've connected with GNE collaborators with the largest cohort, so would already have this categorized and hopefully ready to go. So there still is a lot of paperwork in transporting DNA around the world. We aim to take 3 to 4 families with founder mutations, and here we want to prioritize the unusual cases. So unusual cases are multiple family members, with some family members where the disease has affected them early on in life versus ones where it's affected them later on in life or hasn't affected them at all and they're quite at an old age, or just severity. So some people may get the disease and it doesn't progress as fast as other people. And the question is there any genetic modifiers that are actually contributing to that thing we're observing? The last thing is a pilot study. So we'd like to go ahead with about 12 samples from the NIH to perform whole genome sequencing. So just working with one group, because that will speed up things, and it's a way of actually putting all the framework in place with Maduri's group, with other groups that are wanting to contribute, so they can see sort of how the workflow from start to finish will look like, and how the data will be shared, how the data will be analysed. So that's the thoughts, the progress to date, and how we're going to move forward. I'm going to have a brief stop now before I transition to the other proportion of the talk, and that's looking at biomarkers and creating a patient biobank, to take any questions from the audience if there are any.
Do we have questions from the audience?
Hi.
So no questions at the moment?
Yeah, I have a question. This is regarding the disease model. Do you have any ideas about what kind of disease model would be used for GNE to try and understand? As you said, that it's important to know the disease progression and so on. So we don't really have a disease model. Do you have any ideas about it?
Yes, I think that's a great question. So I think that, so I don't have all the screens on, so I'm guessing that's from Maduri, that question. So yes, I totally agree. We, you know, the mouse models are less than ideal. So I'm not sure. Maybe there are people in the audience that work on some of the GNE mouse models that can better answer the question. And that's why I think we should revisit also the patient cell models and things like that. And another disease I'm going to touch upon that doesn't actually have a good disease model is actually FSHD. And that's why they've actually tried to also work on biomarkers that are relevant to what we see in the patient. So at least when we apply some therapies, not all therapies, that we can look at the behavior of the biomarkers in the absence of a good disease model, because FSHD is another disease where they've struggled to create a good mouse model that recapitulates a lot of the phenotypes, and especially the important phenotypes we see in the human patients, which they can't actually do. But
So does that answer your question, Dr. Bhattacharya? Yes? Okay. We have a question over here from Michaela.
Hi, I was just wondering about your fourth point here about a pilot study. Does it mean you're going to use the samples from phase 2, from the phase 2 trial, in order to perform whole genome sequencing?
We have NIH here that can answer that question. What samples are you using? The natural history samples that coded, of course. Okay. So does that answer your question? Yes, good. Anybody else? Question from Monkol? Does everybody
also,
understand?
I'll just
Is there
add
any
to that.
basic questions?
I think some of the samples we actually want to do for the pilot will be also overlapping with some samples that exome sequencing has already been performed. So it's just a thing that we typically do in these projects. When you do overlapping samples, it allows us to QC, do quality control on some of the samples. So the things that we discovered in the previous genetics project, where we've done some kind of sequencing, do we discover the same things? And it's just, it's good validation data that the whole genome sequencing is actually producing good quality, or as good of quality as previous results that we had from some set of families and samples that we've done in the past.
Thank you for that. Does anybody have any other questions, even basic ones, so that maybe some of this is over some people's heads? If there's anything you don't understand, now is the time to ask, right, Monkol?
Yes, yes, I'm happy to trying to make analogies or trying to make it more understandable, because I don't usually always present in front of the general public. So I'm happy to explain things in a little bit more detail if people don't quite understand, or understand sort of like the rationale.
Right. So I guess that summarizes our, that's it for the questions for us, Monkol. I know you're there in Australia at a different conference, right? Is that where
Yes,
what you
yes.
do?
I'm at a conference, so like focus on discovering more disease genes or genes that are associated with common diseases such as arthritis, etc., etc. But so this is the first part of the talk. I'm going to transition to the second part of the talk, which Angela is kind of like spearheading, but I'll present on it, and then Angela will join us at the end to introduce herself, and also to field in any questions on the second part, or any questions in general from the first part also. So to give everyone ample opportunity to ask any questions they may have that I can better explain, or just to let everyone know also I'm available over email, or could take a phone call if, you know, a question comes up later on, or you're a bit shy, because I'm always shy to ask questions at a conference myself too. So I know the feeling.
Okay, and maybe you could share your Cure HIBM email with everyone right now so they can write it down. And before we go into the second part, is it mlec at Cure HIBM?
Sorry?
Your email, is it mlec at curehibm.org?
Yep.
That's it.
Yep.
MLEK at curehibm.org, everyone. Thank you. Ready for your second bit now.
Cool. I'm going to transition to the next part, and that's talking about, and hopefully this touches upon the first question on alternative ways we can understand the disease in the absence of a really good mouse model. So the measures of sialic acid is currently not a robust measurement. It's a great measurement, but I think there's sort of like room for improvement to reduce some of that variability, because it is a tricky thing to measure, like a lot of sort of protein-based biomarkers, and there are many things that contribute to the variability. And it's the hope that if we have patient cells, we can maybe calibrate these assays a little bit better. Also, some of the challenges, and this is a challenge with all neuromuscular disease genes, even with Duchenne muscular disease, Duchenne, is that patient cells, when you look under the microscope, they look exactly the same as healthy cells. But this is under the microscope, and certain things are measurable, but it's just to have a set of things that we actually can measure. And the reason why we should persist with these biomarkers, such as sialic acid, and also what we can discover in patient cells, is just to supplement the fact that we don't have a reliable animal model for GNE myopathy. It does have a phenotype, but whether that is sort of like relevant to the patient's condition is sort of questionable still. And the reason why we need to have these resources, or to have an improved set of resources, so it may be a better animal model, is that we need to know any therapy we're trying in the patient, if it's actually making a difference. And the reason why is that the disease in many patients is very slow progressing, like limb-girdle muscular dystrophy, that, you know, patients and also the people providing the therapy would actually want to know earlier rather than later if it is actually making a difference. And by working on these things, it will actually help us understand that. So in terms of pathways to therapies, there are already some strategies that have been presented at this symposium or previous ones that relate to gene replacement therapy or dietary supplement of things that the body struggles to make because GNE myopathy is not there. And the clinical outcomes is the long-term, we hope to see an improvement of physical performance and clinical presentation, and overall an improvement of quality of life. So for myself, for me, I would want that. I don't want people to say, hey, your biomarkers look higher, but I still struggle with things that I struggle during my everyday life. And so that is the long-term goal. But the short-term is to see changes in disease marker, because it's the hope that these things will change first before the longer-term things. And therefore, we need to have a good set of things that we're actually going to measure moving forward also. So what is a biomarker? So biomarker can achieve many of these things, but a biomarker is an object that is measured and evaluated as an indicator of normal biological process, or the pathogenic processes or pharmacological process to therapy intervention. So it can be used to detect disease, detect even what stage of the disease, so how the disease is progressing, your response to any kind of treatment. So it's a hope that it's reversed from what we see when we're trying to detect or looking at different stages of disease, and also looks at how efficient it is. So at what degree does it make any particular changes, and also monitor the progression. So it can be used as time points or milestones to actually look at that. So here we want to focus on genetic biomarkers instead of just biomarkers in general, because biomarkers are typically proteins, things that are readily detected in the blood, and also metabolic sort of like properties of things within the blood. So genetic biomarkers can come in two forms, the DNA or an RNA. So the differences between DNA and RNA is that DNA is static and normally doesn't change much during the lifetime of the patient, while RNA, which is sort of like a copy of sections of the DNA, and normally we make copies to actually make a particular protein. And so we make copies and then we make the protein. So this is dynamic. So in, during the day and during your lifetime, different levels of protein are needed. So different levels of RNA is produced, or different expression of RNA is produced. So we've already touched upon the importance of looking at biomarkers, or in this case, more specifically, looking at genetic variants in patients by performing whole genome sequencing. But RNA, we're actually looking at expression. This is actually more dynamic, and it's the thought that there may be expression that's different between healthy and patients, or patients and their unaffected siblings, so their unaffected brother or sister. And this is a dynamic thing, and we believe that this will probably change during the different stages of the disease as the disease progresses also. So I've already touched upon the genome sequencing, and also Madhuri has talked about that. So RNA sequencing will be, the goal of it is actually to create sort of like a disease profile for GNE myopathy. So it's the hope that if we did RNA sequencing, that we can differentiate later on the different, a profile that looks like a diseased patient versus one that looks like a healthy patient. And in a way, you can already see that if we can create this sort of profile, that if we did apply any therapeutic or intervention approaches, we hope for that profile of a diseased GNE myopathy patient will look more like a healthy patient. So I'm now going to touch upon a disease that Angela works on and the benefits of actually creating a good set of biomarkers and how that has advanced their field. And like GNE myopathy, and I've stressed this a few times, they've actually struggled to create a perfect mouse model or even an ideal mouse model. There's another, FSHD is another form of a progressive muscle disease where working on genetic biomarkers has been very successful in establishing disease profile. So by just looking at the expression levels of particular genes, that we can actually tell the difference between a patient and, say, their unaffected sibling. So many biomarkers have been discovered, and the ones in asterisks and the ones listed here are what we call robust biomarkers. So we see them time and time again in patients with FSHD. So sometimes you see some biomarkers that are highly variable. We're interested in the ones that we see a lot of times reproduce over and over again. So they're robust and they're reliable, and then we can use it as a proxy of the disease state itself. So, and here's an example of what you could do if you had disease biomarkers. So in an experiment that Angela did, she discovered drugs and pathways that could be targeted, and this is through a high throughput genetic screen she did to identify a particular pathway. And here in the blue dots, well, here I'm showing results from 3 different genes and their expression levels. And here you can see the expression level of patients that are untreated for a set of drugs. And here you can see the results of the expression level when they're treated by various drugs. And you can see now the expression level of this genetic biomarker for this gene is down regulated or reduced. And now it looks more like the patient, so more like healthy controls. So healthy controls will be at 0. So and that's what we're trying to see. It's the hope that if we see a signal in the biomarker itself, that it will correlate to improving the disease or halting the disease itself. And that's why it's important. And these are actually on patient cells. So it's actually, you could argue, also directly translatable to humans, and what would happen in a human, as opposed to something happening in a mouse. Sometimes it doesn't directly correlate to what happens in humans. So these are the biospecimens that hopefully a lot of you in the audience have volunteered to actually provide. One is blood and one is skin biopsy. So for blood, we hope to extract DNA so that patients can be sort of like considered for the whole genome sequencing that Maduri and her group will be performing. And skin biopsy, I'll touch upon what that could be used for. So the process of the skin biopsy is it's what we call a punch. So this stamp that comes down, a metal stamp that comes down, and it just pulls a little bit of your skin. And that leg there, that's actually my leg. So if you're a good detective, you would have noticed the legs that I showed at the start of the presentation looks very similar to those legs here. And that's because they're my legs. I gave a skin biopsy last year, and it was, I can say that it was a painless experience. They did it in my lower leg, so I can still look good every day. No one actually sees it. My legs are always covered because my legs are quite cold all the time. And so, and you can see the chunk of skin, and this is the back of my head here. And so it was a very quick process. It took longer than expected because I talked too much, and the nurse couldn't do what she needed to do, but it is a very quick process. And there's relatively little discomfort, and the skin heals pretty quickly, and my lower leg does not heal very quickly. And I can say that that was my concern, that it would take a while to heal given my circulation in my lower leg, but it was totally fine. So what we want to do with the skin biopsy is to extract the RNA and also to produce cell lines. By extracting RNA, we can then look at genetic biomarkers. We first can compare the RNA expression between patients and, say, their unaffected sibling. And we can do other things also. And by creating cell lines and having it sort of like owned by the NDF, I think it will create a more collaborative nature and also a more consistent way of a set of cells that have been created and hopefully can be shared with all researchers working on GNE myopathy in a cost-effective way, so not to be a barrier for research. And also, for the blood samples, I already touched upon extracting genomic DNA. So we're looking for affected families and their first-degree family members, siblings. So what I mean by first degree, siblings, parents, or children. And the reason why first-degree family members would be great to have is that for the skin biopsy is that the thought is that the genetic differences, which should be about 50% between them, should be a lot better than just using a random person in the population to compare against. So that would be a lot better. But I'll touch upon another way we actually can use the patient skin cells or cell lines extracted from the skin cells for to reduce the variability. But when we go to the question sections, do ask any questions for people considering or have sort of like promised that they will give that, because it's important that all your questions get answered, because this is a big ask. It's an inconvenience to people that are giving it, because I have given a skin biopsy before. It is an inconvenience. For me, I had to drive an hour and a half to a place that was willing to take a skin biopsy. But yeah, for everyone to know that. So the next steps would be RNA extraction from skin biopsy. So the plan is that the skin biopsies will go to our lab in Yale, where we'll actually extract the RNA and have it there for hopefully enough for other researchers also to use to answer some of their research questions and make that available. And also to be set aside so that we're sometime in the future we can do RNA sequencing and hopefully build upon. So this isn't the first time looking at expression differences between patient healthy muscles. Unfortunately, I didn't have time to show a screenshot of a publication by Sellers Group, who looked quite a while ago at different expression levels using an older technology. But here we want to use the latest technology to see, one, if we can reproduce the same results of Seller and her colleagues, and also to build upon that, is there other things using newer technology that we can sense that we couldn't sense before. But first of all, to reconfirm some of the results that she actually had, and to build upon which sets, particularly the ones that we reproduce, are now considered more robust. And hopefully by sharing this RNA with other researchers, they can also validate this too, because it's always important for multiple researchers around the world to validate the same result. Well, the cell lines, what we first want to do is do sequencing to validate the gene mutation. And it's important for two things. One is to make sure this is a GNE myopathy affected one, or what GNE myopathy mutation. So it's just due diligence. Like with the whole genome sequencing, the first priority would just be confirm the mutation. And the second is, by having this, other researchers don't have to do this from scratch, and they know which mutation they're actually studying. And thirdly, it's to, I totally forgot what the third reason is. I'll come back to it. So the other thing we can do, and this is what researchers in University of Massachusetts Medical School did, was to create muscle-like cells from the fibroblast. So yes, fibroblast is not your muscle, but we have to strike a balance between inconveniencing you, because I have given a muscle biopsy before. It sucks, it hurts, and it takes a long time to heal. So I know exactly how it's like for the patients in the audience also who have given a muscle biopsy, whether it's a needle or an open biopsy, it really hurts. So it's to hope that we can create muscle-like cells without inconveniencing patients too much by taking a skin biopsy. So creating muscle-like cells are not exactly like muscle, but hopefully we can answer some of the scientific questions and understand the disease better with the muscle-like cells, and we can create other cell types such as iPS stem cells with the cells, and then create other different types of cells to study the impact of GNE myopathy in different organs or tissues. The last thing, and I think this is the most exciting thing also, is we can test other gene editing approaches. So I'm going to touch upon the differences between gene replacement and also gene editing. Lastly, the extraction of DNA in blood. I think someone asked this in the audience, is that now we have a fresh set of DNA and it's been consented properly and it's ready to go. It's from patients at this conference that some of them will be actually included in the whole genome sequencing project, because some of the patients that will donate will definitely overlap with the inclusion criteria. And in addition, some patients may have a natural history study, have been enrolled in clinical trials, producing extra information. And lastly, to have the extra information of having RNA and actually cell lines makes it sort of like a fantastic resource for both researchers to understand the questions. Research mechanisms better, and also further questions that may have to have that extra information, and also have that information kind of shared. But to let everyone know that all the samples will be de-identified. No one will know who it actually belongs to. Only a certain set of people, part of a research protocol, will actually know. So privacy will actually be sort of like maintained throughout the project, like most clinical trials and other research-based projects. So so gene therapy, there are two different approaches. So the delivery of a healthy GNE gene, which I've mentioned where we are in terms of the mouse model, that it has been shown to be safe and efficient. So it's being delivered to the tissues that it needs to be delivered to, but we don't know if it's effective. And that's where we are at with gene therapy. And that's the hope of doing the whole genome sequencing, understanding the disease better, understanding what may be modifying the disease, and also creating these patient biobanks so we actually can test some of these things in patient cells and seeing what effects it is before we move on to the patients themselves. Because I think Lali has said this previously in other symposiums and conferences and others, that AAV, because after the first application, your body will recognize the virus, and the second time and the third time will never be as effective as the first time, because a lot of intervention has to happen for that to actually happen the second and third time. And so just to note that. And the second way we could achieve gene therapy is editing the gene mutation itself. So instead of giving a replacement, we're just fixing it. So the analogy is, this is a horrible analogy, but the analogy is, say your car is not working and someone gives you another car. That would be a replacement therapy. And, you know, it's the hope that the other car they give you is just as good and you like that car. Editing is, if your car's not working, you bring it to the mechanic and the mechanic actually fixes your car. So you can drive out the same car. So all the things that you love about your car is still there. So that would be the difference between the two. Both tries to achieve the same thing by giving you a working car at the end of the day, but they do it in a little bit different way. So with the gene editing approaches, there's two broad approaches. The first is cutting out a bit of DNA using this exciting CRISPR-Cas9 that many of you would have heard of. It's always on the news, the biotech news. So one approach would be to cut out the piece of DNA and then replace it with a new piece of DNA. So it's like a cut and replace. If you're using Microsoft Word, you'd be cutting out a bit of text and putting in a new bit of text. The problem with this approach, and this approach has been what's been used over the years, is that it's not very efficient. It's about, the highest efficiency you can get is about 1%. So 1% is very low efficiency for this to actually happen in a patient's cell. The other approach, which this technology has only really been available for the last year and a half, and it only has taken off in the last few months, is being able just to change one letter. So a lot of mutations, as I showed with the founder mutations, they're actually only caused by one letter change in your DNA, in the GNE myopathy gene. So by changing that one letter, you now have a gene, a copy of the gene that's exactly the same as everyone else in the population, so the healthy people in the population. And so it is a long road to get to here, but using the patient's cells, we'd like to actually give that a try. So if we actually make that change, how efficiently can we make it? And actually to screen over, to validate it by doing the sequencing over it. But like I said previously, one of the challenges with working with patient cells with GNE myopathy, at the moment, we can't reliably tell the difference between a patient cell and a non-patient cells. It's the hope that we build upon the biomarkers that have already been discovered, optimize it, or not us, but having that available, it can be optimized by researchers around the world, and that through RNA sequencing, we can actually detect more genetic biomarkers that now we can actually look at the consequences of actually making these changes, such as a gene editing approach or other therapies also. So it's a way to sort of like supplement the fact that our GNE myopathy mouse models aren't perfect mouse models by having these other resources that researchers and people that are aiming in developing therapies, whether it be gene therapy or other therapies, can then use to actually develop, optimize, and make better therapies in the future. So with that, I'll end my talk, and I think Angela can also jump on, introduce herself, and then we can take questions. Cool. So I'm going to pass it on to Angela. Just wait.
Oops. Hi, everyone. It's good to meet you all. Is the camera on?
Yes.
Oh, okay. So my name is Angela, and I'm Monkol's wife. For those of you who don't know me, yeah, I work alongside Monkol in Yale as a research scientist, and I have a lot of experience in understanding rare neuromuscular diseases, particularly in areas of molecular and in cell biology. So we're very excited to be working with the NDF in better understanding GNE myopathy and bringing translational therapies to the field and to patients. So living with my husband, who's been diagnosed with a rare neuromuscular disease, I think we have, I'd like to think we have a unique perspective on what it's like to be both a patient advocate and also a scientific researcher in the field. So obviously we understand the hurdles and the frustrations that come along with having a genetic disease, but we also have, I think, unique insights as to why the science can sometimes seem painstakingly long and a difficult process, and why working together and engaging the patient community can hasten the process. So I think the NDF is doing a really great job in fostering this sort of community that promotes interactions between the patients and scientists. I think right now I'm very excited to be helping with the NDF coordinate the tissue collection and biobanking efforts from GNE patients, as Monkol touched upon in his talk. This resource, I can't tell you how extremely valuable this is to the scientists and the field in helping us make decisions about choosing the right therapies and also in evaluating the efficacy of therapies that are to be trialed. So thank you to those who have opted to donate samples in the form of blood and also skin biopsy, and for working with us and the NDF on this. A big thanks to Lali and Nancy for making this a reality, and also Dr. Perry and his staff for their generous time in helping to help coordinate this on their end. So yeah, if you have any more questions for Monkol and myself, do we have enough time for questions?
We do have, hi Angela, this is Lale. We do have enough time. Just so you know, we are in fact collecting samples as we speak, but in order to make sure they get to you at Yale by tomorrow, we need to be done by 5, and they're taking longer than expected. So perhaps we're going to ask Dr. Shea if he can accommodate some of the local people here after the conference. Are there any questions for Angela and for Monkol? We have a question here from Tara.
Oh, are you going to hold it? Okay. Could you explain the procedure they're going to do with the skin biopsies? More in detail for the patients who are going to have the procedures done.
They're happening right now as we speak. But yeah, could you talk about the size of the needle and the size of the scar? We're getting a lot of questions.
I saw it being done, but Monkol actually experienced this, so I'll let him tell you about that.
So what did you want to know?
Like the size of the needle and just like more details about the process of giving the skin biopsy.
I'm not sure if Perry's in the audience.
No,
but,
he's busy collecting samples.
Okay. He would be the best, but I can say it from being on the other end, so the one that's actually receiving it. So it's actually not a needle; it's just a metal utensil, as you saw in the slide, that just takes a little bit of your skin on the surface.
It doesn't go into deep.
It's about this big, so it's only about half a centimeter. I'm sorry, I don't think in inches. And it doesn't go any deep.
Yeah.
And they do give you a local
anesthetic. Yeah.
So you don't feel any pain. Afterwards, the question is always afterwards. Do you feel pain afterwards? It's no different to a pain where you get a scrape on your skin. So for me, it just felt like that. And, it's done pretty quickly. So it's not a needle. It's more this metal utensil. So there may be people already in the audience that gave
kind of like it takes kind of like a core. It's like a cylindrical kind of device. And it's small and it's really fast.
Well, just so, not to cut you off, Monkol, but I think we've got some patients here who've just done it.
Almost, but it's a lot tinier, like a quarter of the size of like a standard hole puncher.
Angela, can you hear me?
And he could just, Monkol's had a muscle biopsy. That one really affected him a lot. And he was out of action for a while, but with this needle, like a skin, it doesn't go in deep. It's like superficial, it's on the surface. And he could just walk like normal. And Monkol's a really big complainer, and he didn't complain much after this. So I'm guessing it was relatively painless.
Thank you for that. We actually have some people here who just did it this afternoon, and they're telling me that it was It was easy? Okay, here.
Very simple. It wasn't painful? No, not at all. Not the least. I didn't even know that it has been done.
Okay, well, there you have it then. So... Did you all three have it done?
Had it done, and my mom's usually all three of us had it done, and my mom's usually she's I would have never imagined she would ever get a biopsy done of all the things, and she did, and luckily she wasn't looking at it, but she did bleed a little bit, but it was, but that's, I guess the most painful part, if you would want to call it painful, is literally the anesthetic itself. So when at the point it's actually going in, that's when you feel just a little burning sensation, and then after that, it's nothing at all. Like, you don't know when Dr. Shea, who's getting it done, or who's getting everything done for every one of us over here, you don't even feel when the actual instrument pinches into you.
Okay, so we're told he has a good hand. Thank you for that. And this was just moments ago, right? Today, this afternoon. Okay. 5 minutes ago. All right. So if you still feel like you don't want to do it, please don't feel pressured, but you're a baby. No, I'm kidding. You have a question for us as well?
I think it's a pretty basic question, but I was curious on the gene editing. When there's an omission or a deletion, when you were talking about that the new research where they can change a letter, can an omission or a deletion also be fixed, or is that further behind in the research?
So she's saying, can a deletion be amenable to the base editing?
So for the base editing technologies, no, but I showed two technologies, and the other one, for duplications, deletions, they may be possible to cut out that region that's affected, replace it with a template that looks the same. But the challenge with using that method is that it is not that efficient in actually correcting. So if it has an efficiency of less than 1%, then that's sort of like a maximum amount of cells that you actually can affect. So, but the thing about GNE myopathy, like all recessive diseases, everyone has two mutations. So, and if we can correct one of the two, say yes, one of them may be a deletion, the other one may be what we call a single base change or a single letter change. So we may be able to still target the other mutation. And if you can fix 1 out of 2, as far as I'm aware,
it's good enough.
It's good enough
as therapy
because many people already have a sibling with 1 out of 2 of the mutations.
Yeah, and
so
they'll be fine.
Yeah, it's just something to consider that. But if both human mutations are that, then therefore using some of the editing technologies won't be applicable.
Yeah. So if at least one of your mutation is a base pair change, then it should be okay. Both do not need to be a base change.
Okay. Does that answer your question? Any follow-up questions to that? Yes, we have a question over here from Michaela. 1 second.
We know the more the better, but in order to encourage patients, ideally, how many samples would you need?
Sorry, I didn't quite get that.
It was a bit echoey.
She wants to know how many samples you need. And I can answer the question because they've been gracious about it and said, listen, 2 is better than nothing. We have 24 patients signed up to do that today. I don't know that we'll get to everyone, but along those lines, Dr. Tahseen Mozaffar over here has offered that he can also do this after the fact for those of you who are local at University of California, Irvine. Do you want to elaborate on how they can?
Sure. So again, if you're closer to Orange County or maybe San Diego, we can definitely draw the blood. We can even do the skin biopsy. We do the skin biopsies all the time, and I personally have had a 4mm biopsy, which is much larger than what you guys will be getting, and it's really not a big deal at all. It takes less than 5 minutes. So we can work with Lale and arrange for it on a separate day if you guys would rather not do it today. But
we have a question comment here from Marianne from NIH. 1 second.
Hello, Monkol and Angela. This is Marianne from NIH. Nice to see you, and so great to have you guys on board on this exciting journey. Very great initiative, and it's very much needed to start a biobank. And as I said, Madhuri's project to do whole genome sequencing. I think we may need to address the RNA sequencing maybe in a separate meeting at some point, because you may want to compare different cell types. To isolate RNA from a skin biopsy may be different than a muscle type. So you may want to compare it to maybe from some patients a white blood cell pellet RNA. But we can address this in at a separate time. But it's I want to say great to have you on board, and a great initiative. And
cool.
Yeah.
Cool.
So
I'll respond to that.
GNE being highly.
We do understand that. I think the tissue where the expression would be the most informative. We have thought about that, and it would have been great to get different tissue sites such as muscle, of course, along with skin biopsy and say other places. But I think the challenge is also balancing convenience and also patient suffering with things. But what we have seen from the Genotype and Expression Project, which is a project that I did work on also, is that the expression of GNE myopathy, the gene itself is actually highest in skin biopsies and fibroblasts. So it's the thought that hopefully some of the things we discover translates also to the muscle cells, but definitely we do agree that we would need to validate anything, any differential expression in other tissue types such as muscle. And then again, that would require collaboration with your, for example, your group and other groups for them to validate, do they actually see that differential expression in other tissues?
Yeah, but I think initially, logistically, we thought that taking the skin would be the easiest and most painless, and patients would, most likely to agree to. And also, I think, so in addition to the GNE gene being highly expressed in skin over muscle, I think that's going to be an informative measure in testing out various things. And also, with fibroblasts, we can always, there's a technique called forced myogenesis, where we can convert them into muscle cells in the lab. So we have that added advantage of being able to go sort of both ways.
But it
Right.
still would be good to confirm it against real muscle cells too. But I think it's just serendipitous that that expression of GNE is also high in skin, and that's usually not the case with a lot of the neuromuscular disease genes. It's normally just high in muscle and also cardiac muscle, and so of course the heart muscle is even harder to get a biopsy from.
Yeah, so I see why skin is used. It's perfect. I would just want to perhaps suggest to also do the RNA sequencing in the same patient, in some of those patients on white blood cell pellets, just to see how comparable skin expression is with white blood cell expression, because white blood cells have the same GNE isoforms expressed as muscle. However, skin is different. But we can talk about this in a more scientific setting.
Yeah, definitely.
I think, do you have a follow-up comment? She said what you wanted to say. We have a question from Dr. Bhattacharya from India. One moment.
Hi. Yeah, it was just a follow-up. So RNA sequencing would be good for biomarkers, but if we want to also understand the pathology of the disease, I think since you have the samples, would you consider doing proteomic analysis, and especially post-translational modification of the proteins, because those may be more important in terms of disease pathology?
Lala, can you repeat that? It's very echoey.
Yeah, I only got the start of the question.
I only got the start, not the end.
Okay, I'm sorry. I'll just say it again. What I was saying is that would you consider proteomic analysis and also
proteomic analysis.
and also post?
At the moment, that's not on the cards. But if if you have any ideas, and we would be happy to put some of those samples towards a proteomic and try to get some funding for that.
But at the moment, I'm not sure. It's a great question, and sorry we didn't think about that earlier. So at the moment, the skin biopsy will go into RNA later, and I'm not sure that's compatible with extraction of proteins for a proteomic analysis. So I'm thinking of, can we get the most use of the skin biopsy, the precious biopsies that the patients have generously donated, if we could do that. But I'm sorry that we didn't think about that. But the thought is, would it still be sort of like relevant to do proteomic analysis on the cells themselves, because that is another source of protein.
Yeah.
But whether it's too difficult now to retrofit, because I know we do have the skin biopsies, but they're going to go in RNA later, and the other half will be to create cells.
So the entire sample is going to RNA later, or would you have a part of the sample frozen or something?
Oh.
Or
So with the biopsy, it's going to be cut in half. One is going to go into RNA later, and then the other half is going to be put in media, and then to create cell lines.
Okay. So maybe we can talk about it later, and
we could take some, cut the half into a quarter, and then freeze it.
Yeah, it will be sufficient for mass spectrometry. We could do proteomics on that. Yeah.
So the team in India, the GNE Myopathy International people, are offering to help do some part of this. We're going to follow up with that conversation later and keep you posted. We have, I think, a follow-up comment from Madhuri.
Hi, Monkol. Hi, Angela.
Hello.
Hey.
Hi. So the plan within the whole genome sequencing project is to try and do some mass spec work. I think it's going to be critical, like what Marjan said, that what we use to do that work, what sort of tissue type, is going to be absolutely critical, because we could skew the results in either way if we choose the wrong tissue type for that. So I think RNA sequencing, stacking up the results for different tissues, maybe whatever we get, and then doing the proteomic work is going to be critical. But I think it should be done.